నమస్కారం. డాక్టర్ బిఆర్ అంబేద్కర్ సార్వత్రిక విశ్వవిద్యాలయం విద్యార్థులకు స్వాగతం. ఈ ప్రసారంలో
Today we are going to discuss about a very interesting topic for MSc Zoology students from Animal Biotechnology, that is the cancer. This topic we have divided into two subunits. One is cellular aspects of tumor cells and the second one is role of oncogenes and tumor suppressor genes in cancer. The first part, cellular aspects of tumor cells, going to discuss by Dr. Prakasham, Principal Scientist, Indian Institute of Chemical Technology, Hyderabad. Dr. Prakasham will discuss about the few important aspects of this. One is the characteristics of tumor cells, transition of normal to tumor cells, major changes noticed in tumor cells, physiological changes of tumor cells, detections of tumors, characters of benign tumors and malignant tumors. So, Dr. Prakasham.
Good morning. Today we will talk about the cellular aspects of the tumor cells. And a tumor cell is nothing but a part of a tissue, normal tissue, and growing abnormally. And tumor cell may be a two types. One is a malignant, that is spreading nature, it has a spreading nature. And another one is a benign tumor which is localized, which does not spread much. In layman words, tumor is nothing but a swelling due to the unchecked, uncontrolled cellular reproduction. And tumor cell may show specific characters like increased rate of cell division and loss of normal growth control, and it is also associated with the differentiation property, loss of differentiation property, and also does not contact each other, that is contact inhibition. And a tumor cell may be ability to invade neighboring tissues, especially in the case of malignant this one, and it show also metastasis. Tumor cell classification is generally they are done with four major groups according to the body tissue. They call a carcinomas. This is benign in epithelial tissue, and this carcinomas is the cells of outside surface of the body or lines inner the passage. And second type of the cancer is nothing but a sarcoma. This originates from the connective tissue and especially with related to the muscles, bones and cartilages. And third type is a lymphoma. It is always starts with the lymphatic system. Fourth one is a leukemia. This is associated with the blood forming tissue. And what are the cytoskeletal changes associated with the cancer cells includes majorly microfilaments and microtubules are going to change, and the distribution and its activity is associated with the cytoskeletal changes, and these are leading to the altered cell interaction with the neighboring cells, and appearance of the cells also changes, and cell addition property also changes, and cell mobility or mortality will be changing. And the cell addition properties associated with the reduction of cell to cell addition or contact, cell to cell contact, and this cell to cell contact may be intracellular or may be external, that is called extracellular matrix addition. This leads to formation of the large cell mass, loss of the contact inhibition, continuous growth and cell mobility alterations. And the nuclear changes is mainly associated with the shape and organization of the nucleosome or nuclei, and this helpful in the diagnosis of the cancer or what stage the cancer is associated with it. And the next one is the enzyme production changes is associated with the always go in concomitant with the nuclear changes. They where the enzyme secretion changes, and it also helps in the diagnosis of the what is the barriers associated with the malignancy and spread of the tumor cell, how much it is spread and what is the content of the enzymes. And majorly detection of the tumor cells is absolutely there is no method available so far, and diagnosis assessing of the tumor is only based on the degree of malignancy of the tumor. And so far developed whatever the so many methods are developed, however none of the method is satisfactory so far. So majorly now the doctors are depend on the microscopic method, and the severity of the malignance is also detected based on the morphological appearance or degree of malignance, extent of spread. Based on this they detect the things. In practice, detection of the cancer is by the two techniques are adopted. The presently tumor grading is nothing but a study of the measurement of the degree of dedifferentiation is based on the histological and cytological criteria. Histological criteria is associated with the alteration of this tissue structure, whereas cytological is associated with the tumor specific structure. And there is a one more term is called a tumor staging. This is nothing but the study of extent of the spread of the tumor in the different parts of the body. There are again modern methods are developed in the detection of the tumor. One is identification of the proteins, because tumor cells produce the specific or associated proteins which are different from the normal cells, and isolation and identification of these proteins will clearly indicate that cancer is present or not, or tumor is associated with that. For this technique, direct measurement is one of the method, and the second one is the specific antibodies against these proteins which can be detected very easily with a measurable time. And above method is most promising and helpful in proper diagnosis and target specific drug delivery which we are supposed to give it. The cancer therapy, mainly three approaches are followed so far. This is the surgery, radiotherapy and chemotherapy. Surgery and radiotherapy are mostly effective for the localized malignancies, and chemotherapy is effective for a metastatic cancer and certain rare tumors, and mainly in prolonged survival it will be useful. But that is very difficult to get a chemotherapy treatment, because normal cells and tumor cells, when they are using a chemotherapy, the chemicals are also going to affect the normal cells. That is one of the major drawback in the chemotherapy. And benign tumors, general characters, if you look at, they do not invade neighboring tissues, may grow size and create problems like breathing, mobility and circulatory problems. Treated differently by surgery and radiotherapy. Benign tumors specific characters includes resemble original tissue without normal relationship and may arise in most tissues, grow in size but do not invade, can be separated from the normal tissue by a capsule of the connective tissue. That means these benign tumors have a thin line of the connective tissue between the normal and the benign tumor cells. There are unique benign tumors are there, just like bone cortege tumor cells produced especially near the nodules, indistinguishable from the normal cells. Epithelial benign tumors contain all tissue components and what like tumor size and lining tissues of the skin and intestinal tract and bladder, and also contain all tissue components or any type of tissue outside the skin, and looks like a closely packed solid tissue. And even you can look at a mole on your body also is associated with one type of the tumor. And unique benign tumors are just like it also can be a pituitary type, and pituitary gland is another one, and a small gland present at the base of the brain, this is one, and consists of the different types of the cells, produces the hormones. This is the normal function of the pituitary gland. When the pituitary benign tumors may be a single cell type, produce only higher hormone of the single type, or it may be a multiple cell type, produces say multiple hormones, and there is non-hormone producing cells also can produce a benign tumors. Arranged in solid cords in the normal glands, and these are the tumors results in the press of the nervous tissue, leads to the blindness, and severe other symptoms and damage to the normal cells, loss of normal cell functions. Other benign tumors are hormone producing tumors, include thyroid and adrenal etc. And epithelial benign tumors may be a tubular structure made up of one of the two different types of the cells and variable amount of the connective tissue. Example includes kidney and breast cancer. And malignant tumors, these are mainly characterized with the slow cellular abnormalities. This is called dyskaryosis and invasion of the surrounding tissues, and diagnosis is very easy when compared to the benign tumors. And standard cellular characters include increase of the cell number, nuclear size and mitotic activity. Loss of normal cellular arrangements is a variation in cell shape and size, presence of abnormal materials and chromosomes. And tissue diagnosis is nothing but how we can check the tumor cell. This is one term called as a biopsy. Biopsy is normally used. This is nothing but a removal of the one piece of the part of the tissue or wherever the cancer is associated or a tumor is associated, and then fix the tissue with the chemical fixation and observe under microscope. This is nothing but a biopsy. It is essential for the treatment mainly to understand extent of the spread of the tumor, define the area of the tumor and dose requirement in case of the radiation, surgery requirement in possible if one is required to do it. This is only associated with the benign. Malignancy, it is very difficult to go for the radiotherapy or surgery. And sensitivity nature, how the sensitive, how much resistance and how much sensitivity is there, and what are the drugs we are associated with, or hormones or x-rays are useful in treatment of the things.
So, thank you very much Dr. Prakasham, and next we will go for the second subunit of this cancer topic, that is role of oncogenes and tumor suppressor genes in cancer. And this topic is discussed by Dr. P. Kevin Kennedy, Principal Scientist, Center for Cellular and Molecular Biology, Hyderabad. Dr. Kevin discussed about what is cancer, growth and appearance of cancer cells, spreading of cancer, root and causes of cancer, genes involved in cancer, that is the oncogenes and proto-oncogenes, tumor suppressor genes, viruses and cancer. Dr. Kennedy.
Role of oncogene and tumor suppressor gene in cancer. So, today we are discussing about the oncogene. Before I go into the oncogene, let me describe brief about the cancer. Cancer is one of the most common disease in the developed world. Cancer cells are abnormal in their growth and appearance. The picture represents there are two types of cells, the cancer cells which are different in shape, the other one which is a normal cell which is of a uniform size and shape. This is a cancer cell which has a false feet and looks different from the normal cells. Common feature of a cancer cells has a anonymous shape and the different content of the DNA. Cancer spreads step by step. If there is a cancer in a body, these cells pass through the blood vessels and they deposit through their enzymes. They made a hole in the blood vessels, and they deposit in another place and start growing that place. That is called the metastasis. These are different type of cancer cells which is infected with the virus, and you could see the shape of the cells before and after the infection. Root of cancer. How the cancer is formed in a body. Cancer arises from the mutation of a normal gene. Mutated genes that cause cancer are called oncogenes. It is thought that several mutations need to occur to give a cancer. Cells are old, not to function properly, normally self-destruct and replaced by the new cells. The human body contains several cells. These cells has a self-intelligence. That means it is programmed inside the DNA when to divide, when to die. This is controlled by the cell itself. However, cancerous cells do not self-destruct and continue to divide rapidly, producing millions of new cancerous cells. A factor which brings about mutation is called mutagen and is described as mutagenic. Any agent that causes cancer is called carcinogen and is described as carcinogen. The causes of cancer is due to ionizing radiation, chemicals, viral infection and hereditary predisposition. For example, ionizing radiation in the sense gamma irradiation. Any cell is irradiated with gamma or UV light or radon X-rays, they go under a mutation. The mutation change the gene can lead to the cancer. For example, if a thymine dimer is formed, that can lead to a change in amino acid which leads to cancer. Chemicals like asbestos, arsenic, chromium, polyaromatic hydrocarbons, dichlorodimethyl trichloroethane. These are all some chemicals which are all carcinogen. When the chemicals exposed to the cells, these cells get a mutation. Due to the mutation, the gene is changed and the cancer develops. There are some cancers which is induced by virus. For example, hepatitis B, human papillomavirus and Rose sarcoma virus. These are all the viral induced cancers and the genes are expressed and the cancer is formed. Tumors and cancer, cells in tissues overgrow or hypoplasmia. For example, in a mole, you have different type of cells are grown. I mean, that is called benign cancer. Example, moles. But these cancers are not threatening cancer and it is not a harmful. The cells will look normal. But in case of malignant, rapid growth will be there, invade surrounding cells and the deposits at different parts of the body and make cancer. Cells differentiation is usually poor. This is a representative example of a mole and a papillomavirus induced skin benign tumor. Another example, whatever you are seeing in the red color, it is a melanoma which is dangerous. Main feature of benign and malignant tumor: rate of growth rapid in the malignant tumor and benign it is slow. Nature of growth: invade surroundings, benign tumor it expands on some tissues. Spread: metastasis via bloodstream and the lymphatic system, but in case of benign tumor does not spread. Cell differentiation usually poor. In case of benign tumor, nearly normal. Cancer usually involves several genes, proto-oncogenes. In normal cells, code for a protein involved in the stimulus of cell division. If altered, many may be oncogene. Alone do not cause the malignant cancer. Require other mutations include one in a tumor and suppressor gene and viral genes. The mechanism of oncogene activation. There are 5 classes of they are: class 1 growth factors, class 2 reporters and growth reporters hormones, class 3 intracellular signal transducers, class 4 nuclear transcription factors, class 5 cell cycle control proteins. All these factors and proteins are controlled by a gene. If any change in this gene by the aim of mutations, they will create into a cancer. How oncogenes are activated? Point mutation, example KRAS, amplification, example NMIB and NMD2, HER2, new ERB2. These are all some oncogenes which is created by the amplification of these genes. Chromosome translocations, example cMIB and BCR-ABL. BCR-ABL is oncogene and it is through the translocation of the BCR with the ABL gene, it forms the translocation and forms the oncogene. Overexpression due to DNA methylations. Some proteins overexpressed due to DNA demethylation, then it is uncontrolled by the cell itself, then it leads to the cancer cells. Chromosomal changes that the genome can occur. For example, translocation, insertion and loop, chromosomal loop, these all leads to the cancer cells. Nucleotide changes in the genome cancer cells. Tumor suppressor gene, somatic cell hybrids, Kundan's hypothesis, mutation and mechanisms that leads to tumor suppressor gene. Loss of function. Retinoblastoma is a tumor suppressor gene, P53 is another tumor suppressor gene, APC is another suppressor gene. Mechanism of tumor suppressor gene inactivation. Deletion, point mutation, mutation followed by duplication, loss of heterozygosity, DNA methylation, post-translational mechanism binding to DNA viral oncoproteins. virus and cancer. Close to 15% of the population of cancer is due to the virus. Epstein-Barr virus, human papilloma virus, hepatitis B virus, these are all some DNA virus, and HIV-1, HTLV-1, HTLV-2, these are all some RNA viruses. EBV, Epstein-Barr virus, this is another virus, most potent transforming agent. This leads to bucket lymphoma and mesodermal carcinoma. These are all the common disease which is developed by the EBV virus. Human papilloma virus and hepatitis B virus are the very common cancer viruses. Insertion of this human papilloma virus in the cells, they lead to a cancer. Human papilloma virus express the E6 and E7 protein. These proteins inhibits the P53 gene, then it leads to the cancer. As long as the P53 gene is not expressed, not inhibited by the E6 and E7 protein, this cell is normal. It is a representative image of a cancerous cells common in genital warts and most common type of HIV virus found in 70% of the cervical carcinoma. Changes when the cell becomes cancer, transformed cell lines derived from tumors. Most changes in the transformed cell lines are immunological cell lines. Growth in much less restricted conditions, reduced growth factors, less anchorages. Cancer is a common disease, transformable disease which can generate from one generation to another generation through the chromosomes. It also acquired through mutations and it is curable. That's all. Thank you.
I think now you have completed the topic of cancer. Now I want to summarize few important things from this lesson. The first one is the tumor is undifferentiated, uncontrolled cellular growth. There are two types of tumors mainly exist. One is the benign tumor and the other one is a malignant tumor. And tumors are classified as four major groups. One is carcinoma, sarcosoma, lymphoma and leukemia. All tumor cells show cytoskeletal, cell addition, nuclear and enzymatic changes. Cancer usually involves several genes, that is proto-oncogenes and oncogenes. The proto-oncogenes are the genes that code the proteins involved in the stimulus of cell division in normal cells. These normally control how often a cell divides and the degree to which it differentiates. If altered or any mutation takes place, it may form oncogenes. An oncogene is a gene that plays a normal role in the cell as proto-oncogene and promote cancer by mutation and carries the ability to induce cancer. The last conclusion we are giving is that tumor suppressors are loss of function mutations that increase cellular proliferation. Thank you.
ఈ కార్యక్రమాలపై మీ అభిప్రాయాల్ని, సూచనల్ని, సలహాల్ని ఆహ్వానిస్తున్నాం. మా చిరునామా ద డైరెక్టర్, ఆడియో విజువల్ ప్రొడక్షన్ అండ్ రీసెర్చ్ సెంటర్, డాక్టర్ బిఆర్ అంబేద్కర్ ఓపెన్ యూనివర్సిటీ, ప్రొఫెసర్ జి రామరెడ్డి మార్క్, రోడ్ నంబర్ 46, జూబ్లీ హిల్స్, హైదరాబాద్ 500033.